How is it that we find ourselves surrounded by such complexity, such elegance?
The genes of you and me, the genes of you and me, we're all made of DNA. We're all made of the same chemical DNA. We're all made of DNA. We're all made of the same.
Hi, you're watching DNA Today, a multi-award-winning genetics podcast where we explore everything to do with genetics, from CRISPR to rare diseases to new research. We have won the Science and Medicine Podcast Award for many years now. We have hundreds of episodes, and we really hope you enjoy these conversations where we dive into so many genetic concepts. I'm Kira Dineen, a certified genetic counselor and your host. To celebrate the year coming to an end, we are reflecting back on the top genetics and genomics news stories during 2023. So joining me for this conversation are two very big leaders in genomics. Our first is Dr. Brendan Lee, who is a professor and chairman of the Department of Molecular and Human Genetics at Baylor College of Medicine. He's also the director of the Roland and Burden Lawrence Bone Disease Program, Center for Skeletal Medicine and Biology, and Skeletal Dysplasia Clinic. He currently serves as the president of the American Society of Human Genetics. So we're definitely going to be talking about the meeting from this year. And returning to the show is Dr. Eric Green, the director of the National Human Genomics Research Institute at the U.S. National Institutes of Health, NIH. Dr. Green's career has involved directing a major genomics research program that included beginning to end work on the Human Genome Project, which we are definitely going to start our conversation with. And for the past 14 years has been leading the efforts in funding genomic research within the NIH. And he's been on a few episodes of DNA Today, including last year's genetic wrapped episode. So I was like, hey, that went so well. Dr. Green, come back on and bring your choice of people. And so he chose Dr. Lee. So thank you so much, both of you, for coming on and just having our reflection on the year conversation.
Well, it's great to be here, and I'm thrilled Brendan was able to join us as well.
I'm delighted to be here. It's been a milestone year for human genetics, and so we're delighted to share it with you.
Yeah, and I think, as I kind of prefaced at the beginning, we should talk about how 2023 marks the 20th anniversary of the completion of the Human Genome Project, which yielded the first sequence of the genome, really, of completion. And Dr. Green, you've been on the show before to talk about this, especially with the telomere project and all that went into this. What are your thoughts, especially someone that's been in the field a little bit longer than me? What are your kind of reflections on just, hitting that 20-year mark?
Yeah, it was a celebratory year for us. It also was, a thought-provoking year. I don't think any of us especially those of us who've been in genomics for a long time, you do reflect at these milestones, at how far you've come and where you're going. I mean, 20 years is a long time, but it's just truly remarkable when you really think about it. I mean, we celebrate things along the way, the reduction in costs of sequencing genomes, now under $1,000. We celebrate the idea that we've gone from one human genome sequence to now literally millions of genome sequences. And it's really humbling in some regards, but it's also inspiring in other ways, especially when we see all the progression we've made across the full spectrum of activities of genomics, starting with the genome sequence, but leading to genome function, understanding genomic base of disease. And as I know, we're going to get into some incredible accumulating highlights of medical applications of genomics.
Yeah, definitely. And Dr. Lee, your thoughts?
Yeah, I think that, when you think about the 20th anniversary of the human genome, it sits within the framework of just human genetics in general. This is the 75th year of the anniversary of the American Society for Human Genetics. And I think the Genome Project is very much sort of the capstone of all that's gone on. And all of that has led us to today, which when you think about the theme of this year's meeting, I think it's apt and appropriate. It's, many genomes, one humanity, or it could be one humanity, many genomes. They all say similar, but also some slightly different things, and the impact on our world today. And it is driven by, at its core, the understanding of the basic information and sequence in the genome.
And with it being
by the way, Kara, when I was saying that, some of it is almost sentimental. Another point that I really thought about this recently. When I was growing up, when I was a kid, and Brendan was about the same age, I think, the moonshot was a big deal. It was a very inspiring thing to follow because, we put a person on the moon for the first time. And, I was about 8 years old. I was a youngster, and I vividly remember it. It was a very inspirational thing. And I remember many times commenting, even recently, that biology's equivalent of the moonshot, which was incredibly successful. Biology's equivalent was the Human Genome Project. And to be part of that was incredible and wonderful and sort of a life experience. But however much respect I have for the moonshot, it's incredible. They've repeated that effort now, I think 11 times. I think we've put 11 times we've gone back to the moon. The biology's equivalent of a moonshot, sequencing a human genome. We've now done this millions of times. That's pretty incredible when you think about it, that this has now become something that is actually now routine and is now part of medicine. And that's really a real testament to this community of scientists that make up, genomics and genetics professionals.
And that we've done it in our lifetimes. I mean, playing off what Eric was saying, when I was a kid, the thing that inspired me was reading The Double Helix, which was, the sort of milestone. And then to think, all these decades later, within our lifetimes, we are sequencing literally all these genomes all around us.
Yeah, it really is incredible. And I like that analogy of going to the moon was such a big deal and we've only done 11 times. It's like, we've probably lost count just in terms of us in genetics, in medicine, in the world, how many genomes we've sequenced, and even genomes that we've gone back and sequenced. If we've done it the first time and now we're doing it again to, have more depth to that. And be more, confident on those letters to each of within the sequence. And it really is amazing. And I can't even imagine what else we have in store, which we're definitely gonna get to at the end of the episode. I want to pick your guys' brain, just, not necessarily in the far future, but even just like next year. Cause, I should have gone back. Now that I'm saying that, Dr. Green, I should have gone back and listened to whatever you said last year and see if that came true. So homework for listeners to do that. But, Dr. Lee, just talking about you being the president of the American Society of Human Genetics and having your 75th meeting, that it's 75 years in, this was a really big conference. And unfortunately, I was not able to attend. Maybe I can next year, but I heard it was a really, really good one to go to. We could do a whole episode in just that, but what are some of the highlights coming out of that, conversations or just breakthroughs, different presentations that have kind of stuck with you in these weeks later?
Yeah, I think that in multiple aspects of the meeting, we first started on looking at our past and understanding, what have been the major milestones, and we touched on it. Things like the Human Genome Sequencing Project, in terms of the information, things like being able to associate human variation, mutations in this genome with human disease, and arising all from the original structure of DNA. And there was enormous celebration of that. I think, a sobering aspect of it, and this is relevant because this is how I started my year as president, it was to really deliver on a very important project over several years, which was to recognize the harms that misuse of human genetics contribute to in our history. In fact, where there were leaders in our society that, contributed to that. And so recognizing that, coming face to face with that, was a very important foundation. At the core, though, it was a celebration of the present. And, when you think about the types of impact that understanding human variation on disease has led to, in the presidential symposium, we had wonderful discussions and talks about cell-free DNA, how, in fact, one can actually deliver diagnostic information in various contexts, in reproductive health, in cancer, because of the discoveries around cell-free DNA, the DNA that floats in blood can be used for diagnostic purposes, but also recognizing, again, where we have still a lot to do. How do we bring these types of impactful technologies to the broader population to address the issues around genomic health access disparity, something I know Eric and I are very passionate about. And then, of course, the exciting announcements that literally happened at the meeting or concurrent with the meeting, with finally the potential, approval or the recommended approval of things like CRISPR editing of the genome for treating genetic disease, and importantly, one of the first genetic diseases that was elucidated, sickle cell anemia, impacting over 100,000 Americans, and that we can actually bring that technology to bear. So it was just an amazing meeting for looking at our past, recognizing where we fell short and facing up to it, and then really celebrating the present as well as discussing about the future.
And you bring up what I think is my number one, wow, this happened this year, is the FDA greenlighting the approval of the first human drug using CRISPR. I mean, I learned about CRISPR 2012 with that big paper that came out that I refer to a lot on this show. And at this time, I was graduating from high school. And so for me, 10 years later, 11 years later, now having my master's in genetics and being a practicing genetic counselor, to actually see this, I thought, I'd be in my 70s, 80s when this would happen. And I'd look back and be like, I remember CRISPR and talking about all this. But to see it, when I'm only 10 years out, it's just is crazy to me. And multiple listeners, including Kim Enochian, said that was their top genetic story. Do either of you want to kind of explain this? Because I'm just nerding out about it, but we need to give the listeners a little bit more information in terms of, how this is working, why sickle cell. Does either of you kind of want to dive into this a little bit?
Sure. Eric, I'm a therapy geek at heart. I grew up wanting to treat, and I have a need to treat. So I am so excited by this. And, at sort of a 10,000 feet level, it is the deliverable of exactly what Eric pointed to, understanding, the genome. There is no way you can edit the genome, change the genome for good without knowing its basic information, its sequence, and how it associates the disease. So decades of work led us to sort of this current discovery, which is that there are elements in the DNA, in all of our DNA, that control the production of important proteins that constitute the function of red blood cells. And as I think you and the public know, sickle cell anemia is a terrible disease that can be debilitating, especially in terms of pain crises, impacting especially our African-American population, but it impacts all individuals. This therapy, of course, is the first one where basically we can take blood cells from a patient, from an individual, infect these cells and deliver the CRISPR machinery, which effectively edits, or in this case, abolishes a control sequence or a piece of DNA that actually turns off a globin protein, which is usually produced in the field of development period. Now, so basically, we're using gene editing to turn on functionally a protein by getting rid of a off switch. And why is that important? Well, it turns out that when you produce fetal hemoglobin, it can replace the function of beta globin, which is, of course, the culprit in sickle cell anemia, as well as another disease this therapy can be used for, beta thalassemia. So in these really terrible blood disorders, where again in sickle cell you have these pain crises, by doing what is basically a cell transplantation to yourself, a patient, we take the cells, we manipulate it, we turn on fetal globin, we put it back in the individual. It then compensates and basically in the clinical trials, it's amazing. It really basically prevents these what we call vaso-occlusive crises, or these pain crises. So functionally a cure. It is really unbelievable, and the ultimate deliverable of understanding all of the implications and impact of the human genome.
And where we can go from here. Dr. Green, yeah.
Well, before we take that forward, I want to give a contrasting set of thoughts and you'll see how Brendan and I, with Brendan being such an expert in the therapeutic side of the equation, where I spend a lot of time being a spokesperson for the field. One of the things about these advances for sickle cell is so important is for the following reasons. And it is a little bit about managing expectations. For many years, when I've talked about the promise of genomics, one of the criticisms that comes back at me is, well, what has genomics done for sickle cell? Because as some of you know, is that the sickle cell mutation was known before the Genome Project ever started. There were 61 rare diseases that we knew what the defective gene was when the Genome Project began. 61, now it's nearly 6,000. But back then, sickle cell was one of them. And we had the whole Human Genome Project, and I don't think we helped a sickle cell patient at all. And now we had 20 years later, and I would frequently get asked specifically about sickle cell. With all this greatness of genomics and all this greatness of genomic medicine, where are the advances that's going to help a sickle cell patient? And we had to be patient, and we had to manage expectation that these advances need to be measured in decades, not hours and not months. And so I'm so gratified to see this for so many reasons, but not the least of which is because I feel in many ways the Genome Project at its core didn't contribute to it. But as Brendan points out, it's that mechanical understanding of how the genome works and all the nuances that went into that, not to mention the technologies for editing DNA and so forth that have followed since the Genome Project. So I do think genomics now deserves some credit to this advance, even if the Genome Project per se didn't directly do it. And so it's really come full circle in a way that I find very gratifying.
Yeah, and I would emphasize that, while sickle cell, we understood the underlying cause, in fact, about 75 years ago. There's a lot of sort of closure around this. Without the Genome Project and all the studies in genomics, we would not have had the roadmap to actually affect this cure. And so it really is the roadmap to getting there. And it's not just sickle cell. There are so many other gene therapies. One of the things I highlighted in this meeting was this decade has been the delivery and the realization of genetics and genomics in the context of therapy. I had the honor of sitting at the FDA Advisory Committee, which recommended the cure, a very similar scenario to this, what we're talking about, for the first ever in vivo gene therapy for the genetic blindness of the eye. And I sat there thinking, my goodness, gene therapy, we've gone through so many ups and downs over 30 years. It takes hard work and investment by folks like Eric and all of us and the NIH to do this. But we really stick to it, and it's accelerating. I mean, you just mentioned it was a short 10 years between that, discovery of gene editing to this. And part of that is because the Genome Project has been around. The blueprint was there to apply it to.
But Kira, you asked what's next. I mean, and I think the big challenges we're going to see now, it's going to be delivery, right? I mean, it's partially going to be what tissues could we get access to? Lots of ideas about could we do this in such a way that doesn't require something as invasive as a bone marrow transplant or something. So that's the excitement. And you know what? Here's a prediction I'll make is that Brendan and I, being practical physician scientists, will come up with all sorts of reasons why all these things will never work. And the good news is we're going to be wrong. We are going to be wrong, and things are going to happen. Then we're going to sit there 10 years from now saying, I would have never thought in my wildest dreams this was possible. And that's actually what sort of gives me optimism, is how consistently wrong I am when I say, it's never going to be possible. And then boom, it happens. And it happens, as you say, not only in our lifetime, in our careers. It's amazing.
Yeah. I didn't think I'd say this, but yeah, I hope you're wrong in some areas where we go way, faster.
That's what gives me optimism.
Yeah, definitely. And I wanted to circle back to something that Lee brought up earlier. Dr. Lee brought up that talking about non-invasive prenatal screening, you kind of referenced cell-free DNA, and that's my home base. I'm a prenatal genetic counselor. And one of the aspects that we actually featured on the show in episode 224 earlier in the year was about this NIPS, non-invasive prenatal genetic screening, looking at Mendelian disorders. So not just looking at, we started with this type of testing 2011, 2012, somewhere around there. And that was really for Down syndrome. And then we added on other trisomies, extra missing chromosome conditions and sex chromosomes and, more and more and more to the point where we can look at microdeletions and all this. But we're upping the stakes once again of looking at these single gene disorders like cystic fibrosis, hemoglobinopathies, which, pairs well with what we were talking about with like sickle cell kind of spanning out to alpha thalassemia, beta thalassemia, SMA, of being able to do this blood draw while someone's pregnant and saying, okay, well, is the fetus affected by this condition? And kind of taking carrier screening almost to the next level, where we're not just looking at the parental information and saying, well, it could be passed down to the fetus and not having to rely on invasive testing like amniocentesis or CVS. So this is something that I've just been amazed by, that we're already doing this. Dr. Green, kind of thoughts on this? This was one of the features in the article that you guys put out once a year at the end of the year for the Genomic Medicine Year in Review. So this was one of the articles that piqued my interest. And I was like, yeah, we got to mention that. What are your thoughts on this?
So just to make sure our listeners know about this article, so my advisory council to my institute, the National Human Genome Research Institute, we have an advisory council, and we have for many years now, I think it's about 13 years or 12 years, we've had something called our Genomic Medicine Working Group, which is a subgroup of the council and has other members on it as well. And one of the things they do is they scan the literature every month for genomic medicine advances, and then they catalog them. We have a website you can come to where it's all discussed. And then once a year, the group gets together and picks the 10 highlights. And the American Journal of Human Genetics features it in the December issue. So it's, I think, coming out in the next week or something like that. So it'll be out by the time this podcast is probably out. And it's worth reading. It's a very brief article. And indeed, that's one of the featured articles. And to me, as someone who's trained in clinical pathology, so that's I don't practice anymore, but that's my clinical specialty, is diagnostics and diagnostic testing. It's just phenomenal. And, again, every time you think, all right, NIPT is going to be useful for aneuploidy, that's right. You think, and then somebody ratchets it up, and then it's going to get ratcheted up. And there's just no reason we should put a limit on where this is going to lead to, because it's so cost effective as the technology advances, and because it's so powerful. And so I think it's incredibly exciting. And I also think that the activation energy to have, actually, Kira, you're more of an expert than me. You're on the front line of this, but these blood draws are, obviously going to be much more acceptable than more invasive procedures. So the more we could do in these non-invasive procedures, the more we'll be able to have to offer in the long run.
Yeah, and it's just impressive. And we're going to have links to all of the articles we're mentioning in the show notes, so you guys can head over there, or dnapodcast.com. It's impressive that the negative predictive value of this was 99.4%. So for people that them and their partner or sperm donor, egg donor, whatever their situation is, if that comes back that, okay, we have a carrier couple, or, we have this carrier situation where the fetus is at risk, we can do this testing, and you guys are a little smarter than I am, so make sure I'm saying this right. But if it comes back normal, low risk, then we can feel pretty good about, okay, fetus is not affected by the condition, where an amnio isn't really warranted in that case. Am I getting the statistics right?
Yes, I think you are. I think, when it comes to this, it also depends on what you're looking for. So these numbers about positive predictive value and so forth is always impacted by the prevalence of this sort of the condition. And this is where I think the subtlety, and this is really important, as we have these amazing technologies, it's important for us to educate other healthcare providers as well as the public on what the meaning of a negative or a positive test is. Because sort of, the analogy people will often use is, what does a positive or a negative COVID test mean versus a positive or a negative, rare Mendelian disorder test? They mean different things because of that sort of prevalence number of the disease. So yes, but as with always in genetics and genomic medicine, it does depend. And this is where the critical subtleties are. It is enormously powerful, but you also touched on an element, which is these are NIPS. They are still screens. And that's an important point of emphasis in terms of, the practice of the medicine.
And the same applies for cancer applications, with liquid biopsy type circumstances, looking for any evidence of early cancer.
Yeah, and that's one too that I feel like I started seeing that term, like, I want to say 10 years ago or something personally. And then I kind of didn't see it for a while. And then it really came back the last couple of years. So I think that's also an area which makes sense, because we're talking about similar technology here, where we're looking at DNA that has left the cell and we're taking a look at it. So whether that's cancer cells or cancer DNA, whether that is pregnancy DNA, like, we're using the same technology, which I think is really interesting there. And this testing and the articles from Jen Hoskevec, who works for Billion to One, and she is the author of this paper that we're referencing, and she also was the guest on the show. So kind of cool if you want to check out episode 224. And one that I don't know if you guys are familiar with while we talk about Billion to One, it kind of blew up in the genetic counseling space in terms of a lot of people online talking about this, the baby peak test, of looking at the way they're marketing it is discover your baby's traits before birth. So unlock 12 unique traits: eye color, hair color, freckles, teeth, food preferences. There was I want to say uproar in the genetic counseling community of how this could be used in a very potentially negative way that scares me. So, there's just a lot out there, but did you guys see this pop up? I want to say it was a few months ago.
Yeah, no, I'm certainly aware of it because obviously our department has been involved in sort of this sort of screening and, in fact, looking at cell-free DNA, one of the earliest applications for dominant de novo disorders, which is often used in the context of when one detects birth defects. The difference, of course, is that test is not a medical diagnostic test. When we think about NIPS, or some of these other cell-free tests, these are done in a medical context, often because whether, as you alluded to, there's a family history of a disease, or commonly when there is an abnormal finding on an ultrasound, in a pregnancy. I think when you get it into the sort of recreational genomics space, which is how I like to call it, there's an enormous concern, I think, appropriately, for what the information is used for and understanding what it does or doesn't predict. And so I think that's really where, without sort of a medical, it's challenging enough, as you know, you're a genetic counselor, for explaining these really complex principles and what they do or don't tell us. And we just agreed or discussed how they were context dependent. And you move into some of this where it brings up a lot of challenges in the direct-to-consumer space.
Yeah, it's one of these things I do try to monitor what's happening with this because, and again, I and my institute are so passionate about things to get the general public interested in genomics enough to learn enough knowledge about genomics to become fundamentally literate, so that at least not to become genomic scientists, just to become patients, because I think genomics is going to become part of the language of medical care, or already starting to be. It already is. And we just so important in everyday life. So we care a lot about enhancing genomic literacy. And it's like everything else. There's a lot of stuff coming at people every day, a lot of noise. And occasionally there's stories that break through, or there's advertisements or companies. And so I always like them when they catch some attention, but they can be a two-edged sword, because I also don't want it to be oversold. I don't want it to be discriminatory. I don't want it to be unrealistic. I don't want it to be flawed. So it is really, I'm always looking for things that will generate interest and get attention, but do so in without negative consequences. So I do think these are things to constantly be monitoring.
Yeah, I agree with that. I think it's like genetics is cool and creepy and scary and all these things at once. And it's kind of like, all right, how do we balance this? And how do we make sure that people are educated on, I think what screening or testing can and can't do. And, then we start getting worried about how people are using it, which is just always going to be an issue and always something that we're doing. Like the three of us are on here because we're educating people about genetics. So, I think we'll always have a job in that sense. One of the other interesting articles that I came across because of the paper that Dr. Green was referencing earlier is polygenic risk scores and family history being independent indicators of inherited disease risk. I was very surprised by this. Dr. Green, I think I'm going to hand this over to you to kind of explain this, because I'm still wrapping my head around it, and I don't know if I quite fully understand what's going on here.
Well, I'm not sure. I'm not sure the office totally understands. So let's be clear. I mean, I think it's one of these examples where you're taking complicated data sets and you're analyzing them in a certain way, and then you're looking to see whether they're giving concordant answers or not. And each are flawed in different ways, and each are powerful in different ways. And it also, I think, is going to be the kind of thing you're going to want to see repeated in different settings and different contexts, and done with slightly different methodologies, because I mean, as a genetic counselor, you know, family history is a complicated beast. And we all know that polygenic risk scores are, I'm not even sure they're adolescents yet. I mean, I think they're late toddlers.
I was
They're
going to
not
say toddlers, yes.
I mean, we've got a lot to learn. So, I think I found that paper to be one where it just is sort of an assumption breaking paper where you sort of like, just don't assume that these things are going to correlate. They may or may not. And just like, you got to keep an open mind. I really want to emphasize, especially in the case of anything related to polygenic risk scores, there is significant research being done now. And I think there's a lot of well-designed studies that are happening and more that are needed before we fully can appreciate either the power or the pitfalls. And there's going to be both. And I strongly suspect that we are seeing an uptick in the literature that's going to accumulate on a whole host of issues related to polygenic risk scores, not the least of which, of course, and some of that is discussed in some of these papers, are all these issues around proper use of polygenic risk scores from different populations, different ancestral groups, and the biases of current genomic data.
Yeah, I think there is a lot more we have to learn there. And I like how you phrase that. It's more just remember that just because we have certain polygenic risk scores doesn't mean it's exactly going to line up with family history. Like there's so much more to explore here. That's definitely not a final statement on that end.
Right. It's interesting. All of these approaches are different levels of resolution. I think that family history, as practicing geneticists, and Eric and I will agree, they're the ultimate sort of readout of environment, genetics, variation at all levels, and differently in different families. So perhaps at some level, while I agree, it was surprising at first when I saw that paper, when you really think about it, maybe it's not shocking because polygenic risk scores looks at a certain aspect of everything that family history really is a product of, but at a much greater resolution. So they're similar and overlapping, but quite distinct in when you really get at sort of what's being measured. So, but Eric is absolutely right. It underscores the importance of, again, doing the research to understand what it does and doesn't tell us. And that's at the end of the day, the most important principle, and I'm sure you agree as a genetic counselor, of everything we do in genomic medicine.
Yeah, it is. It's like sometimes I feel like, okay, I'm doing another family history, but it's like, yeah, that's still, I think, the best genetic test at this point, definitely could fight me on that. But, as we're learning so much more, it's still an important piece of the puzzle with everything. And another topic that we explore a lot on the show over the last couple years, I'd say, is pharmacogenetic, genomic, looking at all of that and just how this information can reduce serious adverse drug events, which, I'm not as much in medicine as you guys are and have been in your careers, but just of the research that I've done and just being exposed to people that are pharmacists, that this is, I don't know at what point it ranks, but a contributor and sometimes like a common cause of death, of having, just drugs mixed in the wrong way. And unfortunately, we hear this quite often. So seeing that on our side, genetic information can help prevent that is great. And I think that's another area that past 10 years, we've had a lot of, strides in, where I think when we started having these pharmacogenetic tests, especially in the psychology space and like looking at what SSRIs someone should be on, like, years ago, it wasn't so great. And now it's like getting much better. So I think that this is just encouraging that we're making a lot of headway in this area, I think, as opposed to, other years. I think we've seen it much more. Would you guys agree with that in terms of the strides that we made this year as opposed to other years in pharmacogenomics?
I still think, and Brendan may have a different opinion, I still think it's facing a glass half full, glass half empty kind of area. I mean, there's a lot of reasons to believe that it should be further along than it is in clinical practice, and it hasn't quite gotten that traction. One of the things I'm going to be looking for, and I think it's still a little too soon, but maybe we'll see, is, to me, the lack of uptake and the lack of definitive evidence has been for a variety of reasons. But I tend to think, or I guess I'm hopeful, that the way the All of Us research program has been organized that the amount of information that might be forthcoming that includes a lot of historic medical information about individuals coupled with genomic information, coupled to real-time capturing, that maybe that will give us the kind of strength of data in terms of sheer size that might move the field forward. That's just a hypothesis. I don't know if Brendan agrees with that or not, but I still feel like that there hasn't been a sort of enough data in one place to really look critically at some of these things.
I think, it's interesting, this topic, because it underscores the title of the presidential symposium at this year's ASHG meeting was Delivering on the Promise of Genomic Medicine, Not a Sisyphean Task. So I think the reason I named it that is because some might think it has been a Sisyphean task, and I don't think it is. I think pharmacogenomics, as Eric pointed out, is exactly that, the half cup full versus empty approach. And, there are lots of reasons for it. And I think the paper that was highlighted in the annual sort of review touched on it. There are economic reasons, which are an enormous driver, obviously, in our health system. But then there's real sort of questions about effect size, and what is that effect size? What's the impact of that finding? And that also may be different and is changing in different health systems and practices. I think I remember when I started my career, the electronic health record was about to start, and the prevailing 99% consensus it was going to destroy medicine. And I think it has to do with the inherent sort of practice patterns that American medicine especially is sensitive to. And I think one could do a whole psychosocial analysis of that topic, because there's so many variables that has impacted on why it hasn't or has not actually delivered on that promise. I think, again, in certain contexts, it clearly does. And I think, but there will be research that continues on what are the barriers for implementing it. And there are a lot of things that are both scientific and not scientific about that.
So this is one to keep our eye on and kind of see how it develops and how much it really is going to become part of our healthcare system, of like, okay, look at either a certain panel or look at someone's whole genome and kind of look at certain variants within that, say, oh, these drugs are, a better fit for you, or this dosage is better fit for you. That I hope in my lifetime we get to that point of having that help us. And also, as this article that we're referencing, reducing those serious adverse drug reactions too, of like on the other end of things, of just the more serious. Maybe we'll see that before we see mainstream tailoring of medication. But yeah, that's interesting you say, of just, starting EMRs, EHRs, like of people saying, oh, this is going to ruin everything. And it's like nowadays it's the opposite. If you're paper-based, that's very challenging and has its own set of things that people are like, it's, living
so
in
I'm hopeful.
age.
That's the bottom line. It's within our lifetime. That seems to be the theme to this afternoon.
Yes, hopeful. And I think the last story I wanted to mention, and then, happy to hear about if there's other ones that we haven't mentioned you guys want to talk about, but I'd be remiss if we didn't go full circle and talk about whole genome sequencing, and especially for critically ill newborns. This has been a topic that, we've, I think, explored, I'd say, the past 2 years on DNA Today, and also the host of the Phenotyp Speaker Series. So, we've talked to some prominent geneticists in this space, like Dr. Kingsmore at Rady Children's Hospital, that, has a big role in this in terms of just looking at, okay, these are newborns that are critically ill. And if we're able to get genomic sequencing back in, hours, days, we may be able to save this baby's life by getting a diagnosis and changing treatment. Dr. Green, thoughts on this? This study was looking particularly at natural language processing and helping to prioritize these newborns. But, I was kind of taking at it from a little bit of a global scale there.
Well, I love the example. I feel it's almost a little personal to some extent. 14 years ago, I celebrated my 14th anniversary as the NHGRI director last Friday. So the very first workshop we did after I became director of the Institute was a workshop to look at whether we should have a program to look at genome, the sequencing of newborns. And we put out a program that's since has stopped, called EnSight. And one of the grantees was Stephen Kingsmore. And one of the things that directly came out of our program was the first examples of rapid newborn sequencing.
And this was 14 years ago when you started, because happy belated. I saw that on LinkedIn this morning.
Right. But the thing I like about that story is when I became the director of NHGRI and I pushed a very aggressive agenda for doing more research in genomic medicine, and that was one of my first programs that we started. Some people thought this was bad. Some people thought it was too soon. It was not, medicine was not ready for genomics, blah, 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 blah. And the fact that, that became, I think, a home run of that first program that we had of newborn sequencing. And what has happened since has been, I think, phenomenal. And we're seeing uptake of this around the world in many settings. It's fantastic. And I feel very gratified because I think it really did reflect it was the right time to start pushing, and it ended up being something that we were able to see successfully implemented. And now, as you may know, and I'm sure if you had Dr. Kingsmore, he talked about this, we have government and insurance companies both getting very interested in this. And, that's not always the case in all nooks and crannies of genomic medicine, that people want to see this paid for and they want to see it done, in an appropriate fashion. I think this is a great win for genomic medicine.
I think, there's no question that genome sequencing and rapid genome sequencing, no matter how you define it, one could quibble about whether it's 24 hours or 3 days. And again, that's going to be practice and environment dependent. But rapid genome sequencing is transforming care. And this is why, which is really great news, as Eric pointed out, there are states paying for this already. And I think increasingly payers are, I think, recognizing the value of this. And what NHGRI has supported over the years has directly helped to really bring this to fruition over the past decade. So absolutely, it's clear. Rapid genome sequencing has had an enormous impact and will continue to do so.
Yeah, I definitely think so. I think that's a topic we're going to be constantly coming back to, and having it be in the NICU totally makes sense of that's a great place to start with this rapid sequencing. But then where else are we going to go with it in other areas of medicine, I think is what I'm excited to see, because it's we see records breaking for how fast can we get these rapid genome sequencing done. And Dr. Ewan Ashley, I think for at least a little bit held the title, maybe still does for 40 something hours. I don't know. Correct me. But it's exciting. It's those are the records that I want to see broken, of how fast can we do this? And getting those answers and saving these babies' lives. It's remarkable. Especially when you look at, coming full circle now of being 20 years from the first genome, human sequence. And now we're doing in hours when it took us
13 years.
13 years.
13 years, or at least, to be fair, sometimes 13 years is not the right number. We were actively sequencing human DNA for 6 years.
Okay, but still, 6 years versus 40 something hours. Yeah.
$1 million to less than 1,000. I mean, it's incredible.
It really is incredible. And we're not talking about a century later. It really is wild. But any other stories that, obviously there's plenty of other stories, but others that you're burning to share before we kind of wrap up with some predictions for 2024?
I will just, I could think of lots, because
I know, right?
So much way cool going on in genomics. I did want to not really point out that another thing I think that happened in 2023 has been, even though it was part of 2022 as well, but we are accelerating our development of a pangenome reference to bring more diversity and representation of different human populations into genome sequence references, which is going to be critically important for everybody to benefit from medical advances in genomics. And I think it's just becoming a very mainstream and very routine to be constantly thinking about diversity aspects of genomic medicine implementation. And I just want to highlight that. And because and what I'm talking about not only is genomic medicine implementation, but even the more basic science aspects of generating reference sequences and telomere to telomere sequences. And there'll be a whole science that we need to develop on how to properly use pangenomes and how to, when you have individuals of blended ancestry, how do you analyze their genomes relative to pangenomes? So this is going to be the new complexities we'll deal with. But there's no longer an issue of if there's going to be good references for all ancestral groups. Now it's just a matter of when we'll get it to the point where we have hundreds and hundreds instead of just dozens and dozens.
I cannot believe I left this off of my list. Thank you so much. I would have listened back as I was editing and been like, how did we not talk about Pan Genome Project? Yeah. Because I think, Dr. Green, you and I talked about this on episode 182, and I think at last year's Wrapped. That's when you came on to talk about telomere to telomere, so I think we got into that a little bit. But yeah, that's such an exciting area, especially for, I don't think we talked about it enough on this episode, of just increasing the diversity in the populations that are in our research databases so that we can have more accurate variant curation there. I think is just
but also the references. I mean, the really important thing is to get really high quality references from multiple different individuals representing human populations, so that people have and then figure out, because there's, again, and we're doing supporting a lot of research, we need to figure out how to use those references maximally, but we really got to find the methodology, develop the methodologies and match it properly to the available pan-genome information. And I think we'll squeeze a lot more information that'll be highly informative for genomic medicine in the long run for people of all backgrounds.
Yeah. And I think that also kind of ties into going into 2024, because we're continuously working on this and having this be a major part. Dr. Lee, anything else that that are major misses?
So I think that, I'll play off what Eric touched on in terms of the pangenome and the T2T. What this has allowed us to do, of course, is more accurately, with great resolution, identify genomic changes that in some way could be associated with the disease. So what I would emphasize is the functionalization of the genome. That's currently happening, and that will be what will be tomorrow and the next year and beyond, which is sort of using and applying technologies. And there have been papers from in the past few years really underscoring that things like other omics, like for instance transcriptomics, RNA sequencing, will be important, not only research tools, but diagnostic tools for functionalizing the variants that the pangenome and the T2 assembly will allow us to identify more accurately. But there's a lot of those variation that is associated with disease that's not going to be easily interpretable in the coding part of our genome, and we will need to functionalize that. And that's going to be, I think, the exciting thing that's already happening now with RNA sequencing. It will continue in the future, but that's a deliverable. That's how we get from a diagnostic rate, of 25, 35, 45 to much higher. And I think that's going to be key.
Yeah, I'm excited to just see how much we learn from this. And as you said, using that information to then be able to actually get more results and those diagnoses on there. And I think I have to shout out as just something that, is not just impacted, well, maybe as it will impact genomics and genetics, but just impacting my own life and how I do this show and everything, is artificial intelligence. I mean, I think this was a major thing just in general, taking away genetics, but I want to say that's the buzzword term of the year. I would put AI on the front of the time. There's always a time person of the year. I would give it to AI this year if I had my own version of that. And I think the listeners would agree to some extent, because our top episode of 2023 was episode 231, talking about ChatGPT and AI in genetics. So I think that is what people are learning about, wanting to know. And I am very excited and intrigued to see how we're going to start using AI. And we already have started, but how we're going to really be putting this into workflows much more, of just using AI in genetics and genomics, whether that's variant curation, whether that's report writing, of you get a lab report, are we using AI to start writing some of this and combing through data and so much. I mean, that's just off the top of my head, but I mean, AI has completely changed
return of results.
Return of results. Yeah. I mean, there's just so much that I think that we're going to see in the next decade, but especially next year. Is this year, everybody's kind of starting to get used to ChatGPT and all this. And the next year, it's kind of going to be much more normalized, I think. So I know technically ChatGPT launched, a year ago. So November 2022, but I think people didn't really know about it till this year. But yeah, I would say AI is what I'm kind of not predicting necessarily, but going to be on the lookout for how things have changed there. Any other predictions before we close out? You guys have just been awesome. I think it's been very hard to pick and choose what we're talking about here. This is not comprehensive. It's kind of just us taking an hour to reflect.
My prediction is that 2024 will bring at least 3 to 5 genomic advances that I'll just hear about them and I'll shake my head and I'll just say, wow, I did not see that coming, or I didn't see that coming so quickly.
And that's what we'll talk about next year.
AI realm. I mean, it could be in any of the realms of what we talked about. But
yeah.
I'm just telling you, I mean, I think it's about, I think it's about, every other month. It just seems on average, something gets reported. And I just shake my head and I just, wow, either I didn't see it coming or I didn't see it coming so quickly.
Yeah, we picked a good field. We don't really get bored, do we?
Yes. No.
Well, when I recruit trainees and residents and fellows, genomic medicine and genetics is the only field where we do something different from 6 months ago.
Yeah.
Could be dramatically different.
Right.
And how many fields where, an interpretation of a specific variation can change one day to the next. We certainly don't get bored here.
No, definitely not. And I'm sure listeners are not bored listening to us and everything. This has been just a fantastic episode. I really appreciate Dr. Green, you coming back on once again and fitting me in your crazy busy schedule. And Dr. Lee, it's just been wonderful to get to know you on here and just pick your brain with all of this. As we've mentioned, you can check out the Genomic Medicine Year in Review 2023. We're going to put a link in the show notes. This episode has come out today on December 8th. So if the article is not dropped by today, I will go back into the show notes and update it. So if it's not currently there because you're listening to this episode right as it comes out, I will go back and edit that. But also it will be at dnatoday.com. Thank you both so much. This has been an exciting year. I always look forward to this episode of just being able to recap and talk about things that maybe we didn't have time to fit a whole episode in over the course of the year. But listeners, we really look forward to knowing what you want to hear on DNA Today in 2024. So please email in info@dnatoday.com or interact with us on social. So thank you so much, Dr. Green and Dr. Lee, for coming on. This was fantastic. Just what I wanted it to be.
Well, happy holidays.
Happy holidays.
Yes, happy holidays, everybody, and happy New Year. We still have some episodes of DNA Today left in the year, but we had to get this out close to Spotify Wrapped coming out because this is when everybody's talking about it. So awesome. Thanks, guys.
Take care. Bye.
Thanks for watching DNA Today. To access all of our episodes, head over to dnatoday.com. We also have a lot of bonus content on there that you can enjoy. If you have any questions, comments, suggestions, guest pitches, you name it, send them in to info@dnatoday.com. We'd also really appreciate if you could take a moment to rate and review the podcast on Apple, Spotify, wherever you listen to the show. It really helps more nerds like yourself find the show. Also, if you like giveaways and other ways to connect with us, I recommend following us on social. We're at DNA Today Podcast. We also have a Patreon if you want to be the most level involved in the show. That's also at DNA Today Podcast. Thank you so much for listening and watching. Join us next time to learn and discover new advances in the world of genetics.
We're all made of the same chemical DNA. We're all made of